Okay, hi everybody. For good and bad, this is the last session of the meeting, which is called Plant Physiology and Mechanisms. So I think that one thing we can all agree on is that plants are amazing organisms. And as a scientist, we are inspired by the large repertoire of sizes and shapes that they have, the incredible developmental plasticity, and the fascinating solution plants have evolved to survive an ever-changing environment. In this session, we aim to highlight the importance of understanding the fundamental mechanisms that enable plants to achieve these remarkable abilities at a scale ranging from the single cell to the performance of the entire plant. Such knowledge, we think, is critical to achieve the goal of this meeting, which is to improve crops at the face of climate change. So we heard really great examples of how this basic understanding is important, and in this session you will hear more about it from three speakers that mainly revolve around the physiological mechanisms. So the first speaker of today will be Menachem Meshalion. He is from the Hebrew University. He's an expert in the regulatory role of plant aquaporing in controlling the plant water balance. His research revolves around improving agronomic plant vigor and enhancing yield production. He's also interested in development technological advances in agriculture. And he will talk about discoveries regarding the impact of stomata dynamics in plant response to water regulation. The second speaker is Tamara Zulay-Shemer. She's from the Agriculture Research Organization that is located in the north part of Israel in the Veyar Center. She's an expert in photosynthesis and water balance, and her lab explores plant responses to stress, including temperature extremes, drought, and rising CO2 levels. Tamar is also a leader. She leads a fruits tree sciences unit at the Israeli almond breeding program. In this context, she will present today her research aiming to understand the physiology and the genetic basis for stem photosynthesis capabilities in wild almond, which is a trait that is not present in the cultivar ones. And last, we will have Lisa Ainsworth. Lisa is a research leader of the UCSDA ARS Global Change and Photosynthesis Research Unit, and a professor of plant biology at the University of Illinois Urbana-Champaign. She's an expert in photosynthesis responses. She applies integrative approaches from genetic to agronomy to address crop responses to global atmospheric change and potential solution to mitigate climate change through agriculture. She was awarded the National Academy Sciences Prize in Food and Agriculture Sciences, and she was elected a member of the National Academy Sciences recently. So today she will present her research efforts to identify the physiological determinants of yield change in response to elevated CO2 and ozone, as well as strategies to improve canopy architecture and photosynthesis. So I'm happy to invite the first speaker, Menachem Meshalion.
So, we all know this already, right? So drought is a major impact, and the big reason for this is transpiration. I mean, the only reason plants are so sensitive to drought is because they lose so much water. And the water loss via the stomata in molecular ratio is about 500 to 1 on average on croplands in Israel. That means the plant loses 500 molecules of water in order to gain 1 CO2, which basically make a very bad water use efficiency for crops. And when you think about it, this is not the status at wild type. In wild type, plants lose much less water, but breeding, which was meant to increase yield, also increased the leaf size and the number of stomata and the time the stomata is opening. So when you look at almost any crop, you see they transpire much more water. So the productivity is going up, but also the water is going up. And this is a very nice presentation. We also saw similar yesterday. This is the release of the bread. This is wheat. And you see the yield growing up. This is what we expect from breeding, right? But the same researcher took the same crops and then he tested stomatal conductance. And you can see that as more the plant produces, the more time the stomata is opening. This makes actually the plant, the crops, to take a lot of risk because they use the water a lot in order to get CO2, right, to open stomata. Now, stomata is a fantastic organism. In this slide, you see only the receptor of light wavelengths, right? From UV to far red in a single cell. So plant or guard cells sense the wavelength, the intensity, temperature, CO2, and of course, ABA. When ABA goes up, it's actually overrule all the other signals. So it doesn't matter how light, temperature. If ABA goes up, everything closes down. So we have multi-signals, and when I think of water balance, I think about this kind of a balance. It's a very multifactorial balance that it's very hard to maintain, right? So water balance in plants actually involves many factors of light, temperature, humidity, soil water content, light intensity and quality, and so on and so forth. So maintaining this at a balance means that the plant have to get by with whatever the environment gives them. And also, there are many strategies to respond to these lights, right? So many sensors, so many receptors. Probably there are also very different ways of responding, like in management ways. And this is exactly my research. We are trying to understand what is the management decisions of the stomata. Some stomata open very fast, close very fast, responds to light, don't respond to light, respond to CO2 faster, slower. And we believe that these tiny micromanagement differences is the reason that some plants produce a bit more and some plants produce a bit less. So what we want to see, so we also talked about this before, so breeding for abiotic stress is a very, very long process. And one of the reasons is this dynamics of the plant response and the dynamic environment. So our hypothesis was that if we can screen back plants that were already bred in the field and we already know their production, both under field conditions and in the lab, both under well-irrigated and drought conditions, maybe we can find some of the key physiological traits of this stomata, and maybe by understanding these key traits, we can also use this as traits for future selection. So this is exactly what we did. We took Professor Dani Zamir's tomato introgression lines. The nice thing that all of the plants are M82, but each cultivar has one piece of a genome from the Pinelli. So the plants are very similar, both genetically and morphology, but they have a very different behavior. Dani Zamir was generous enough to give us all the 10 years of field data, both from well-irrigated and drought. When I say drought, it's drought in Israel, so plant will die in drought of Israel. So these are irrigated, but controlled irrigated. So we talk about mild to moderate drought, not a catastrophic drought. So we took all of this data and we screen all the plants, and we look for plants that behave better than the M82. And we look for several behaviors, like you can see here. This plant, this 5-1, actually has always better both under drought and control from the M82, so we call it idiotype. And I want you to focus now only on this 5-2, the green one, and the red one, because we have many, so it will be simpler to follow. I will show you all the results, but it will be simpler to follow on these two ends. And we took another one, which is 8-1, which was always lower in productivity and much more susceptible to the drought. Now, And we make sure we had at least 4 to 5 years of experimental from the field, so we know we can maybe a bit trust the data. How to screen the plant? So there are many ways. We all know these cameras. I don't think these are very good for physiology. Today I'm going to talk about a screening that's called functional phenotyping. I will explain more. But I decided to bring some paper we released lately as editorial in the Journal of Experimental Botany, where we decided to set up some guidelines of how to do controlled drought experiments in pots. So why me and 4 of my colleagues decided to write these guidelines? Because we get many papers on drought, a lot of them. Most of them have some critical issues of just in the experimental design. I'm not going to go too much through it because I want to show the result of the screening we did, but some of the principles we follow. So we call it the 4 Ds. You need to define your stress, try not to go to very general, like we want to screen for drought resistant, blah, 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 but try to be very quantitative when you do it. When you develop your hypothesis, try to use the right vocabulary. I want to give you some example. When you have the stress, so usually stress started and then the plant could die, so it's susceptible to the drought, it's not sensitive to the drought, because if the plant were sensitive, you would probably sense it ahead and respond to it by either escape or other mechanisms. Now, if you respond and survive, so this is a survival plant that actually passed the stress, maybe rain come, maybe we irrigated. And then there is the resilience or the recovery. So resilience is the recovery from the stress. Now, all of these, or even this plant could be considered as resistant if we talk about ecological resistance, because the plant did not die. But from the agronomical point of view, this plant actually has a low tolerance and a low resilience, because it just survived. This is not agronomical trait at all. I'm not going to go to all the details, but also resistant could be only plant that look exactly like control in the yield amount. So if we talk about agronomical point, we usually talk about crop and not just, many times you see in pictures that people show you, okay, this plant did not die. So from agronomical point of view, that doesn't mean a lot. So how do you do this screen? I will talk later on the phenotype, but now I want to talk to you about some big artifact or the major artifact in pot drought experiments. So one of the biggest artifacts is comparing the stress. When you want to do stress for many plants, you want to make sure they're suffering from the same stress. So let's look on a simple experiment. We have a pot, let's say we have 500 ml of water, we irrigate it, and then we let the plant transpire. Let's say the plant transpire 140 ml. So the next day you have only 360 ml, but it grows a bit, so it transpires a bit more the day after, 150 ml. So the next day this plant has only 210 and then 90, and then we see some wilting because the plant doesn't have water to transpire. Now let's say that in other point of the table, when there are only 1 degree more, this plant transpires only 110. We don't know it unless we measure it. That means that this plant will have much more water than the other plant, let's say at day 3 or day 4. Now when we compare the plants based on the day, we actually compare them to different stresses. So taking day in account, especially if you want to collect the RNA or other metabolites and comparing the plants based on the day without knowing the transpiration, this is a big, I would say, biased. So this is how the introgression lines that we work with looks like if you just close the tab here. This is the soil water content. You see some plants almost don't lose the water content and some lose it very fast just because of transpiration differences. Okay? And it could be from the environment or from the genetics. So a small plant or a small transparent plant could be here, and the higher transparent could be here. So of course that after a few days, if you just collect the data here, you are not going to get the right comparison. How do we control it? We control it via special controllers that actually irrigate the plants. We use the plant-based system and we irrigate the plants based on their own transpiration. So we have a feedback system that brings back water to each pot separately based on their own plant transpiration. This system also has covers both from the bottom, so nothing leaks either from the bottom or from the top. Each plant has its own controller, soil sensor, VPD and light sensor, so we know, and irrigation system. So each plant is being irrigated separately based on its own transpiration. Each plant sitting on a lysimeter, so we know the water loss rate for each plant separately. And in the end, this system enables us to reduce the noise coming from a lot of things in the greenhouse and get pretty much clear signal. Because each plant has also its own controller, we can build a truly randomized experiment because we don't need to split the pipes. Each plant getting the irrigation based on its own transpiration. So each plant, actually, we can control the irrigation. We measure several physiological impact, and all the plants are exposed to the same environment. So we make sure they have the same environment and the same soil water content. So they are actually really exposed to the same potential all the time. And we measure all the plants simultaneously and continuously via the drought and the recovery phases, where all the plants sees all ambient conditions. So this is what we call the functional phenotyping. We actually measure continuously the plant biomass gain, transpiration, water use efficiency, stomatal conductance. I'm not going to talk today about root influx and the drought point. I will show you this result soon. And it's absolute measurement, so it's not like a porometer or Likert that you measure a piece of a leaf, you measure the whole plant. So let's go back to our screening. We want to compare the plants. So this is 8.1, you remember the poor looking or the poor behavior plants. So this is day by day, this is the daily transpiration, how much water in gram the plant transpires per day. And you can actually see the plant growing. We don't see biomass here, but we see functional trait. When you look on the idiotype, you see it transpire much more, and I want to remind you, this plant produces more even under drought. So this is a big surprise. How come a plant that loses more water in the end produce more? I'm going to, and this is the M82, this is like, let's say, the mother plant somewhere in the middle. And these are all the other plants. I don't have time to go now to details, but we can later, if you want, can discuss the difference between all the lines. And this is the soil water content. So all of them under the soil water capacity all the time. We make sure the water are available at this part of the experiment, no drought at all. Then we expose the plant to drought. Look how nice the drought look. Because we use the feedback irrigation, so it doesn't matter if this plant, you see, it's lose twice than this, but the soil water content go in the same rate because the feedback irrigation. And then we have a recovery phase, so the plant allowed to recover, and we measure how much time it takes the plant to go back to its previous transpiration. You see it's few days, but you see a big difference between the line. Personally, I believe that this recovery is one of the most important traits to define drought response. Because if the plants activate many mechanisms here, it's all going to benefit here. During the drought, you hardly see anything. There is one thing you see, and I will show you soon. So we compare the drought and the recovery. Now, what is drought? Okay, what is stress? This is, again, it's a physiological point. So we define stress not based on the time, but based on the soil water content, the start limit transpiration. So on the y-axis you have transpiration, and on the x-axis you have the actual soil water content. This point here is where the water content in the soil become limiting factor for transpiration. To me, this is the physiological point. No other traits really show you this limitation like the water content that start limited transpiration. And of course you compare all the plants on the same trait and look for the different trait. But when you want to compare them under drought, it's the best to first to be after the tetacrit, but second under the same stress. So we choose a phase that all of the plants were under similar stress and below the tetacrit. So we know they are stressed, the closed stomata. Okay, and we try to make them as in similar stress as possible. Again, it's very important when you compare apples to apples. So what do you get when you measure this? Now I'm going to the resolution of minutes from day to minutes, so you can see if we take the transpiration rate, this is transpiration rate, and this is stomatal conductance. I'm not going to get to details now, but this is a very critical point that actually calculate the stomatal opening, but not based on looking, but based on the calculation. Stomatal conductance, like your porometer gives you. So this is the 8.1. It's look nice. But when you look on the 5.1, you see very interesting response. You see that in the morning, when the VPD is low and the light is sufficient, they fully open the stomata, although I want to remind you, they are now under 50% drought. So this plant has dynamic water use efficiency where they open stomata at a certain time of the day, much wider than the other plants, and then they close it down. This transpiration shows the actual water loss. So this behavior allows them to lose less water, although they open stomata wider, but only like 1 hour a day. Is it enough? Probably yes, because in the end, as I show you, they produce more. So this 1 hour a day probably accumulates through the season and allows them to, in the end, produce more. So they're under the same stress, but behave differently. We did it to all our lines, and we wonder why would we have this different stomatal conductance behavior. So we went and we looked at the stomata. We just used this resin that you put on the leaf. We did it 8 times per each line during the day in a greenhouse because we want to reflect as close as possible to the measurement, to a real world measurement. And we did it to the abaxial and adaxial leaf, so both bottom and top. And you see very interestingly that the total density, the gray line, was hardly different between the lines. But, so if you take both top and bottom, no significant difference. But when you compare the top to the bottom, the 5.2 has much more stomata on the bottom of the leaf than on the top, while the 8.1 has a similar ratio between the top and the bottom. When you look on the stomatal aperture, okay, we really measure it in the microscope, you see that the 8.1 fully open stomata at noon, which is not a smart thing to do in Israel. While the 5.2 only outdo it in the morning, as I showed you before. So what the system shows is really reflect the real stomatal aperture. And I want to remind you, it has much more stomata on the bottom of the leaf. So theoretically, you know, the same line, M82 just cross, and you see totally different stomatal pattern and stomatal behavior in a different time of the day. So, you know, the difference is in the small details of the behavior. Of course, we did it to all the lines, and we did another thing. We look for different expression for both speech. It's a gene that regulates stomatal development, and the ZEP, which is actually related to ABA production. I will run a bit faster here. But what we found is very interesting. There was a nice correlation between the speech expression and the number of stomata at the abaxial part. And the opposite for the ABA. So the 8.1 has much more ABA probably. We didn't check the ABA, only the gene. And the 5.2 was the lowest. So it seems like ABA is responsible to the dynamic behavior, and speech 1 is for the number of the stomata in both sides. We didn't prove it, it's just a correlation. We also, at the end of the experiment, we cut the plants, we dry them, and we check the biomass. So you can see clearly also on the lysimeter, this 5.2 and has much better biomass, just dry biomass, after 4 weeks of experiment. And the water use efficiency. Water use efficiency, it's agronomic water use efficiency, which means we take the dry mass of a plant, And because we know the total transpiration of this plant throughout the experiment, we divide the total ml, it's in the gram ratio, of the plant with the total dry matter, and then we get agronomic water use efficiency. It's actually agronomic transpiration use efficiency because we cover the soil, so it's just the transpiration. So to sum up, what are the traits, the physiological traits that we think that are important for a better stress response? You see, I don't like the word resistant or tolerance. It's just a better stress response. So first, fast growth, so when the water are well, if the plant grows fast, it's just better, more leaf, more photosynthesis. Then, very high water use efficiency. So this fast growth is actually translate well to also nice absorption of CO2. Fast stomatal closure, so when the drought stress, they respond and close stomata. Much higher concentration of stomata, or stomatal density higher at the lower part of the leaf than the upper part of the leaf. This golden hour, we call it, so using the daily VPD and light in a way that will be much more efficient for the plant, so open the stomata when the VPD is low. So we call it dynamic water use efficiency. And recovery. As I told you, recovery was fantastic because this plant transpired almost twice in this. And even though you can think that the stress was more severe to it because it's actually a bigger plant, bigger leaf, but then it recovered faster, and this is like 4 or 5 days. So this resilience or recovery, I think, is one of the interesting traits. Okay, so I just want to thank my collaborators that helped me writing the 4Ds paper. Sanbon Goza was the PhD student that make actually most of the experiment here. Professor Dani Zamir actually gave me all the data from the field. Without it, we cannot do it. Bogala, Ravi,